I'm Dr. Richard Visconti, and I know stem cells. I'd like to tell you today a bit about the historical background of stem cells and their current application in medical science as well as basic science research. So scientists have studied and known about stem cells for a considerable amount of time. However, in the last decade or so, stem cells have received new attention for their potential to be differentiated into cells and tissues that can be reintroduced to the body to treat pathologies and illness. Historically, stem cells have been studied for their contribution to development as well as how they contribute to postnatal tissue repair and regeneration. Embryonic stem cells, which are found in very early developmental stages of the embryo, have the ability to give rise or differentiate to become every cell type in the body. Whether we're talking about fish, puppies, or humans, embryonic stem cells are able to divide and differentiate into all of the cells that make up that organism. Postnatal stem cells have been known for a considerable amount of time as well. Perhaps most well known and characterized are hematopoietic stem cells. These are the stem cells that are found in the bone marrow. They are able to divide to regenerate themselves. They also are able to differentiate to give rise to all of the cell types that circulate in the blood. So from a clinical aspect, these postnatal stem cells have been used in the clinic for decades, particularly for patients who suffer from blood cancers like leukemia. So if you have a relative who has been treated for leukemia, they generally undergo chemotherapy and perhaps radiation, and then their bone marrow is repopulated by bone marrow cells, hematopoietic stem cells, that have been harvested from a donor who is a close genetic match to the patient who needs the bone marrow replacement. So with the advent of molecular biology and genetics, stem cells have been rediscovered for their potential to be differentiated into cells and then tissues that can be used to treat disease. So whether we're talking about cells from the embryonic inner cell mass or from postnatal sources, scientists have now begun to differentiate these cells in a petri dish into cell types that they would like to make, whether it's neurons in the brain, cardiac muscle that is found in the heart, skeletal muscle, pancreatic tissue, basically any cell type that they like. And there has been a large amount of success with being able to direct the differentiation of stem cells into these cell types. Subsequent studies using animal models like mice have shown that this ability of stem cells to differentiate can also be followed within the animal so that cells and tissues can be directed to differentiate by implantation into a particular organ of the mouse, meaning that stem cells can be injected into the liver and evaluated for their ability to listen to what the cells are doing around them and become a similar type of cell. Another application for stem cells would be to direct their differentiation in the petri dish and reintroduce them into the mouse model as a differentiated cell type. And that's to say, the cells are differentiated into liver cells, for example, in the dish, and then reintroduced into a mouse whose liver has been damaged, and then these transplanted cells are evaluated for their ability to repair or regenerate that damaged tissue.